I'm at Shaldon Wildlife Trust, and they've been a long-term partner of the Little Fireface Project, in fact, for about 12 years. Shaldon Wildlife Trust is the smallest zoo in the UK, but they have a huge number of very endangered species that are small, including slender loris, Bengal slow loris, and the northern pygmy slow loris. And here you can see that this zoo has won a number of awards from the British and Irish Association of Zoos and Aquaria. And they do a lot of amazing work with small conservation projects and also a lot of work with enrichment of their animals here in captivity. And we were really lucky enough to win a few of these awards with our partner zoo, including an award on the accelerometer project, as well as an award on conservation education between children in the village where we work in Indonesia and children here in Shaldon village where they were pen pals with each other discussing their views of conservation. So it just goes to show you how a very small conservation project and a very small zoo can work together to do amazing things.
Hello, my name is Professor Anna Nekaris, and today's talk is about the taxonomy of slow lorises and why this is important for their conservation. I will be covering several topics, including why taxonomy is important, going over the history of slow loris taxonomy, the current species that are recognized, as well as their conservation status, also suggesting potentially some new species that may be recognized in the near future, and then summarizing this and considering what are the steps forward that need to be taken about conserving these species. So, why do we need taxonomy? Here's a wonderful image from the early 20th century of two slow loris species and two slender loris species. Neither of these was recognized for over 100 years, but now with genetics and other work, we know that these are indeed four species. And you're going to be learning about two of the slow loris species today, and very large issues that could affect their conservation. So first, one of those issues is simply understanding the geographic range of a species. If we don't know what species we have in our hands, for example, confiscated from illegal wildlife trade, we may reintroduce species into the wrong country, the wrong range, the wrong habitats. And that means that we cannot protect habitats that are important for that species. Perhaps we have two species that at the moment are considered one that have very different habitat needs. But since we think there is only one, we neglect to protect one of those habitats. This has direct implications to their conservation status. A species that is restricted to a small range may become endangered or critically endangered, but if it's thought to have a wide geographic range, it may be considered least concern or near threatened. So again, it's incredibly important to just understand where these species are. Species needs vary. Why is this important? Here we see just a little sketch of several of the slow loris species. You could already see they differ by body size, they differ by color, which probably has something to do with the forest types that they're found in, which may have important implications for sleeping sites, for habitat continuity. And of course, this could have implications for captive management, the amount of space needed, the amount of food they are fed, the type of habitat that is managed, how it's managed, what type of connectivity is left. Of course, it also is important for education about these species. So a number of slow loris species do very well in human-modified landscapes. but it's presumed by some governments and in some countries that they can only live in a pure forest. So species may be moved from neighborhoods or rural areas that where they're living in people's home gardens and put into forests where they don't know how to find food or how to thrive. So it's very important to know in each case, in each country and each population, what the species needs are. There are also similarities that may occur even if there are different species. So this is just one important point amongst the slow lorises, is they are a group of animals that are hyper adapted to excavate, gouge for, consume and digest tree gums and exudates. This is very important, not only again for keeping them in captivity, for understanding the types of trees that they may need replanted, because as I said earlier, they may be very common in agricultural landscapes or human landscapes, or even in areas where replanting is being undertaken for corridors, etc. It's not the type of food item that may be used by many other species. So considering planting gum trees may not be so important, but for slow lorises as a group, it is. And here you see the very important tooth comb that allows them to actively gouge, which is something few other primates can actually do. Home ranges may also vary via species and via the habitat type. Here's just an example of pygmy lorises with quite a large home range of 12 to 22 hectares, being one of the smallest slow loris group of species. It varies very much from the Javan slow loris, whose home range may be as small as 6 to 9 hectares. And here we have in the middle the slow loris Nycticebus kukang from Malaysia, where the species was studied more on a beach habitat and more in a forest habitat, and just the habitat type itself changed the amount of space these animals needed from 3 hectares to 25 hectares. But even at that smallest number of 3 hectares, it's a lot larger than what many authorities think lorises need during translocations. So again, knowing this variability among species, among body sizes, becomes very, very important for managing these animals. Where should we survey for them? What type of area should we protect for them? And here we see known habitats of different slow loris species in Java, India, Cambodia, and Thailand. And it's very, very different, very variable. And it doesn't mean that slow lorises can exist everywhere. It means the species differ, and it will, and thus it's important to understand this. So how many slow loris species are there, and why does it keep changing? People get frustrated with me and my team when we want to name, or we do name, a new slow loris species. But we are by far and away not the only group doing that with nocturnal primates. And we've named far fewer species than most people working on nocturnal primates. Here are some examples with the mouse lemurs, which have dramatically increased the number of species that are recognized. The tarsiers not only have dramatically increased species, but their genera. Same with the bush babies, which more and more species are being found all across Africa. And here you see the East African galagos, which have undergone a genus name, being many millions of years separate from their West African small galagos, which look very similar. So even in this slide, a lot of these small nocturnal brown primates look very similar. And that's because they don't need visual systems to tell each other apart. They have different mate recognition systems. This may come from their smell, their calls, the shape of their sexual organs, and it often takes genetics to tell these species apart if we don't find, for example, that identifying different calls are enough. Genetics simply haven't been done for slow lorises in the wild. Illegal wildlife trade itself has confused genetics because lorises have been moved around and reintroduced or translocated into the wrong places. And even mislabeling the samples that we have on GenBank, for example, or mislabeling of museum specimens makes things very confusing, because in a period where everything was called Nycticebus kukang, and I'll explain how that happened in a moment, it means that a lot of people don't know what they're looking at, or they're lazy to know what they're looking at, or the loris was sampled as part of a larger study on primates in general, was put into some sort of a system like GenBank as the wrong species, and it compounds the problem. This is also incredibly important for reproduction. I mean, obviously, if you have multiple species in captivity, sometimes like this animal you could see here, this was a hybrid from Singapore Zoo, but the hybrid itself wasn't fertile. We also can have invasive species impacting the native species. There have been examples of hundreds of slow lorises of the wrong species being reintroduced into the wrong country where other slow loris species existed, and reintroduced onto islands where slow lorises never existed. This could not only cause problems for the native species of slow loris, but in the case where no slow loris ever existed, the slow loris may become a competitor for another type of animal that's in that habitat if they happen to survive. So, it's not as easy as it seems. Slow lorises are incredibly affected by illegal wildlife trade. They're frequently confiscated. They're frequently in rescue centers. They're frequently even purchased by well-meaning citizens who want to just translocate, reintroduce them back into the wild. And most people just see them as a brown, small animal, and it's very difficult for them to hone in onto the features that actually make the lorises look different. And because, let's make this very clear, the appearance isn't that important, even myself as an expert, sometimes it's very hard to tell these animals apart, especially like what you see here. You see a very young animal, and the young animals can look even more like each other. So when did the first loris, when was it described, and what is the journey happening now about slow loris taxonomy? We first get a description of what we now know is a type of Nycticebus bengalensis in the 1770s by Vosmeer, who was a really important collector of different specimens. He named all sorts of species. And lorises were initially a little bit confused with sloths. And here we have pentadactyl sloth, the 10-toed sloth. And it kind of went into his collection until it was named as a species in 1812. So there we see an early representation of Bengal slow loris. It's a very large slow loris species, more or less with a whiter head and a lighter body, sometimes a brown body. And below that, you see a Javan slow loris with a very funny old 19th century representation of it catching a bird. We do know these guys catch birds, so I like that this early representation catches this rare and unusual behavior. And it was named also in the early 1800s as a very beautiful specimen. And if we look along the lines, in the 1800s into the early 1900s, we had a number of taxonomists and natural historians traveling throughout Southeast Asia, naming new species. And we could call this more or less a series of splitting, because if they found a species that was smaller, had a different cranial or skull morphology, that was living on a remote island, that was living in an area that seemed to have geographic boundaries, it tended to be named different species. So here we have on Java, two species, Nycticebus ornatus and Nycticebus javanicus. We have Nycticebus pygmaeus, which is found in Indochina. In fact, there were two more of these as well, Intermedius and Sinensis from China. Then we have a series of Bengal slow lorises, Bengalensis, more or less from India, Cinereus, more or less from China, Incanis, more or less from Burma and Myanmar, Tenasserim, more or less from southern Thailand. This transfers over to Malayanus, a type of kukang. And then we have Nycticebus kukang here in a couple of subspecies, kukang, brachycephalus, and hilary. And then we have a number of the Menegensis types, Borneanus from Borneo, Menegensis from Borneo, Philippinensis from the Philippines, Natunaensis from the island of Natuna, insularis from some of the Malaysian islands, and Bangkanis from Bangka and Belitung. So there are some others around as well, but you could see a lot of splitting happened. And here we could see, if we put some circles around them, they were divided nowadays into 1 or 2 or 5 species. With this, and some of them being confusing. Is it a Meningensis? Is it a Kukang? But we will talk about that more in a moment. So in 1953, the incredible primatologist Osman Hill wrote a series of tomes, essentially looking at the taxonomy and systematics of primates. And this was a period of lumping, and he took all of those slow lorises and he lumped them into a single species of Nycticebus kukang. We then go fast forward almost 20 years to 1971, when Colin Groves entered the world of primate taxonomy, and he started to write a series of papers. In his seminal paper on lorises, decided that Nycticebus pygmaeus was different enough to be its own species, retaining all of the other lorises as subspecies within kukang, and again reducing the number of subspecies as well. He reconsidered this in 2001, and he really felt that indeed the Indonesian forms or the insular forms were quite a bit different than Bengalensis, which has a distribution from India all the way into China. Its cranial morphology was different. It's significantly larger. It regularly overlaps its range with Nycticebus pygmaeus. So he thought, let's go for 3 species. We then have a genetic approach by Christian Roos, who wrote his PhD thesis in 2003. And he was looking at strepsirrhines in general, and the lorises were amongst this. And he found, with some material from museums, from illegal wildlife trade, that there was evidence for 5 species. So here we start to get a more modern, what we're starting to see now with the slow lorises, with again the Indo-Chinese forms of Pygmaeus and Bengalensis. Then we get Meningensis more or less on Borneo and the Philippines, Javanicus only on Java, Kukang in Sumatra, Singapore, and we get them in southern Malaysia and Thailand. And it's a little bit easy, but within this we start to see still more difference of these animals. And this comes to some work that I did with colleagues Munz and Ford, where we looked at the face masks of these animals. And we also went back to the original descriptions of the animals. We looked at biogeographic boundaries. We looked at species that have been recognized of gibbons and macaques and langurs. And lorises, of course, evolved earlier and dispersed throughout Asia earlier than the monkeys and apes. And they are more restricted by their locomotion, by the biogeographic boundaries that also would have separated monkeys and apes. Yet we still don't have nearly as many lorises as we have monkeys and apes. But we did find evidence for a number of species on Borneo, including Bangkanis, Borneanus, and a new species, Nycticebus cayenne, as well as separating Hillary from Nycticebus kukang. So a bit going back to some of those earlier descriptions. The only newly named species here is Nycticebus cayenne, which is largely in the west of Borneo, but we don't know much about its distribution range in the wild. When we actually go on to do a bit more of this with museum genomics, we find, in addition from our face masks, we find some confirmation that there are 2 species in Sumatra. We find that there are at least 3 species on Borneo, but the distribution we had from face masks doesn't perfectly match the distribution we find from these museum specimens. This doesn't surprise me because, again, it's not just about how these animals look. They're very similar looking to each other, but there are geographic boundaries that prevent them from mating with each other. So if they look different and we have these geographic boundaries, this is getting to be pretty powerful evidence. Most important is that we had evidence for the pygmy slow loris being a distinct genus, separating more than 10 million years ago from each other. And we also got strong genetic evidence that there is a northern and southern form that are two distinct species. So this goes back again to the early days where 2 or 3 pygmy slow lorises were recognized by early explorers. Here you just see a phylogenetic tree showing the separation of northern and southern pygmies, northern, western, and eastern Borneo, 3 types of Bengal slow loris, the peninsular kukang really deeply separating from the other kukang. And you see a couple of Javans in there, but I'm not surprised by that Javan in the north because many people mislabel Javan slow lorises into Bengal slow lorises. This suggests to me there's just another Bengal slow loris out there. So we go on to the IUCN red listing. We had the first IUCN red list, which I was the main author of in 2008. I was involved in the next red list workshop in 2015. And then we were re-asked to look at all of those in 2020. And we have 2 of our species as critically endangered, the Javan slow loris from the island of Java, the Bangka slow loris from the island of Bangka, possibly Belitung. At the moment, all the Bornean forms were listed as vulnerable, but this is based on virtually no ground truthing of data in the wild of how those populations are doing, considering most other Bornean animals are considered primates are considered endangered or critically endangered, and all of the mainland forms and Sumatran forms are listed as endangered. So let's discuss the differences between the slow loris species. Nycticebus kukang is the one that everyone knows. It's called Kukang Sumatra, the greater slow loris, or the Sunda slow loris. Its distribution, you can see here, is in southern Sumatra. We don't actually know the dividing line between these 2 species, between it and Nycticebus hilary, which is in the north of Sumatra. It's also found in southern Malaysia, in Thailand, and Singapore. And there is an incredible variability in this species. It ranges in size, a 300-gram range, 450 to 750 grams. It's can be a little bit more brown. It could be a little bit more red. It tends to have a flatter face. I have to say that the southern Malaysian form looks the most different, and it was at one point a different species. I often say with this species, it has pointier circumocular patches. The muzzle is much smaller, and it looks really cute. It's like a cute ball. It tends to be a bit rounder, but it's you'll see the difference between this and some of the other species. Here we have Nycticebus menegensis, and it is called Kukang Kalimantan. It is also called the Philippine slow loris. The subspecies from Natuna is the Natuna slow loris, and I like to call it the blonde bombshell, the beautiful blonde slow loris. It's found in Sabah, in Sarawak, in Malaysia, in Indonesia. It seems to be found all over the island. It seems to be a species that can be sympatric with other species on Borneo. Of course, it's in Brunei, some of the surrounding islands like the Natunas, and it also does occur on some of the westernmost Philippine islands, where again, it was originally called a different species, and in the Philippines, it tends to be the blondest of all. So it's very different than Kukang in the fact that it's much smaller, and it's got a lighter fur. It weighs less. In fact, from museum specimens and from some work done in the wild, it is the lightest of all the slow lorises, and it's also got a really small body length. And here we start to see, using the face mask data, how other slow lorises were separated. And at the top there you see Javan slow lorises. In the middle there you see Kukang with Nycticebus hilary, and there you see menegensis at the bottom. And this Nycticebus hilary, that is very, very different, has a very dark face mask. It's the one you see here. And it has a longer muzzle, and the crown of the head almost looks like a person with 2 arms, 2 legs. And it has a very dark patch around the eye. If we look at this new species, Nycticebus bornianus, they are very, very fluffy. That's the really incredible thing about this species that makes it different. It's not, we do get fluffy fur in juveniles and subadults of slow lorises, but in the case of the bornianus, we have fluffy adults. And just to make this point more clear, this is the first time that I saw adults of what we would call Menegensis and bornianus in the museum in Chicago, the Field Museum of Natural History. These animals were collected from a similar locality around Kota Kinabalu in Malaysia. They were collected around the same season. They were both adults, or all 4 of them were adults based on their dentition. But you can see the incredible difference in the coat of the fluffy bornianus versus the very slim and long Menegensis. Also, using those face masks and looking at the museum specimens, these are from Naturalis in the Netherlands. We see this incredibly beautifully deep red crimson loris, which is Nycticebus bancanis from the island of Bangka, also potentially from the island of Borneo. We now have a rescue foundation on the island of Bangka working a bit on rescue and release of these animals. And we finally, for the first time since the 1930s, have photographs of them. And they are just as beautiful in life as they are from those museum specimens. Nycticebus cayenne, which we know does occur in Sarawak and Sabah, and it should occur in other parts of Kalimantan. The main difference here really is this incredible facial mask. So it's not fluffy like the bornianus. It's although it is still fluffier and darker than what we would call the Menegensis, but we have this face mask that extends all the way below the cheekbone, which is very distinct and different either from bornianus or from Menegensis, and it is larger than Menegensis. For bornianus, we don't really have any body weight measurements. We move on to the Javan slow loris. For a while, people thought there were 2 or 3 species of Javan slow lorises. This drawing that was made of a dark form or typical dark form and a light form was done by a colleague of mine, Helga Schulze. But in fact, this dark form tends to be more the juveniles, and at one point was put into its own species ornatus because it was so dark. And the lighter form tends to be the adults, and I'll show you some images. These are only found on the island of Java, and they do seem to be distributed east to west. Their main distribution is to the west. There seems to be a gap somewhere here in the Middle East, and we get some in the dry zone here in the east. Again, we're not sure if those animals were always occurring in East Java or if that's due to translocations, because there is a big trade in Javan slow lorises going on to the island of Bali, where they do not occur. So who knows? We don't know. We don't have historical records from the east. And it is a very different habitat for the Javan slow loris. So whether or not they originally occurred there or were translocated there is still remains a question. This is a subadult Javan slow loris with the darker eye patches. And this is something that we see in these animals, that their fur is white and dark brown. They can weigh around 900 grams. Older skinny ones or young subadults, young adults, maybe around 800 grams. Pregnant females or really large males may get to 1 kilo or 1100 grams. But overall, the average is around 900. And there you see the body length. If we look at the facial masks of a very, very young juvenile, then we go to a very young adult, a slightly older adult, and a nice old lady there. We can see that it changes incredibly. So their face masks change as they age. So when we're using something like color, it's not going to be a straightforward measure of species. It may also be a measure of individuality and of age. So the species I haven't talked about yet is bengalensis. This is the most wide-ranging species, occurring all the way from India to China. It weighs 650 grams to nearly 2 kilograms. And just looking at this image alone, you can see stripes and no stripes, white fur and brown fur, crimson fur, forked heads, non-forked heads. And early on, these were a number of species which are now called bengalensis. The closer they get to their overlap range in southern Thailand and Malaysia, with Kukang, we do see maybe we have a hybrid zone, maybe we have a transition zone, or maybe we just have another species there. And work by Hiroko Somira, who is a geneticist working from specimens confiscated in illegal wildlife trade, suggests we have multiple species in Thailand. And the work we did with Mary Blair with museum specimens and museum genomics suggests we have several bengalensis specimens across their range. So more work needs to be done here. And where could we look for bengalensis boundaries? Here, if we just consider gibbons, which also look very similar to each other, despite being a diurnal ape, their differences are actually less than the differences we're seeing in the lorises. And using genetics, we now have 16 species and 3 genera across that same range. We get the slow lorises. So there would be no surprise if there are more slow lorises occurring there. Here we have a new subspecies, Insularis. It's unclear when the genetic work was done because of this complication in GenBank, if it's a Kukang or a Menegensis. It does really look more like the Menegensis type or even a Bancanis type. And we see another island form with this bright red fur. But it is recognized now on the IUCN Red List as an endangered species, subspecies. And here you see another form that does not have a taxonomic difference from Langkawi, but a very dark form that complies with a general bengalensis shape, and it would be considered bengalensis, but it could be a new species. We also get the pygmy loris here. So the pygmy lorises, as you know, they were the first species by Groves to be re-separated in 1971. And in fact, when we look at the genetics of these animals, they are 10.5 up to 20 million years diverged from Nycticebus. And when we look within them, in the northern part of their range, in the southern part of their range, going into Vietnam, not going into China, where a third species was initially recognized, we do get 2 genetically distinct species, which also have pelage differences, with the southern pygmy loris having shorter pelage with less coat color change seasonally, the northern form changing color in the cold winter months, with a fluffier pelage, definitely going into torpor, no evidence of that in the south. And in the south, we have a longer muzzle that's also evident in the skull, and a shorter muzzle in the north. So a new species and a new genus. So this is just to remind you, nocturnal mammal taxonomy is not straightforward. Nocturnal mammals are generally brown. They don't use colors to tell each other apart. So when we see these big differences, it's even more of a sign that they could be species. By not recognizing that these are species, we can do great damage, having hybrid species, invasive species, before we even know which species exist. Where we find species boundaries of other primates especially, we probably can find new loris species too. And what's incredibly important is that this is taken seriously, that rescue centers and captive management programs manage their animals appropriately, that they don't just feel exasperated or stressed that a new species is named and ignore it because they can't tell a cryptic nocturnal species apart. This also is important that governments accept these changes in taxonomy in their endangered species law. And at this critical time where we're losing enormous amounts of habitat, it's better to recognize more species than fewer. In the future, when things may be more stable and forests are preserved, we can always put them back together again. But if we have this genetic and morphological and behavioral evidence that we have more than one species, It hurts no one to, at this time, preserve these populations as unique taxonomic and ecological units. Thank you so much for listening. I hope this was a useful overview of slow loris taxonomy. We do have on our Little Fireface Project website at nocturama.org a more comprehensive identification guide that should help you to identify the currently recognized species. You can follow us on our Instagram, on Twitter, or X as it's now known, our Facebook page, and you can also see a number of our ethogram videos, project videos, etc. on our YouTube channel. Thank you to all our funders who have been funding this work for a number of years. And thank you for listening.